Good morning, everyone, and welcome to our second guest lecture for this month. Today, we have Professor Sourav Bhattacharjee here to talk about oral delivery of insulin, a mission impossible. So that's today's topic. Let me take a minute to give a brief introduction to Professor Sourav Bhattacharjee. So he joined the Medical College and Hospital, Kolkata in 1997 and graduated with an MBBS in 2004. After a brief period of residence training in orthopedic surgery, he joined the MSc in biomolecular sciences, cell biology at the Vrije University, Amsterdam in 2006. So in the meantime, he moved to Edinburgh, where he worked for almost 14 months under the supervision of Professor Vicky Stone at Napier University. Upon finishing his master's, he began his PhD at the Virginia University, also in Netherlands, in 2008. He successfully defended his thesis in 2012, and the outcome gives so many research papers with good impact factor. Following that, he worked for a year as a postdoc at the University of Twente in Netherlands. And in March 2014, he joined with Professor David Brain group at UCD as a postdoc working on the EPFP7 funded TransInst consortium, trying to develop nanoparticulate drug delivery platform for oral insulin delivery. From February 2016, he joined the UCD School of Veterinary Medicine as an assistant professor in veterinary anatomy, while also trying to develop his niche of research encompassing a broad range of nanobiotechnology and advanced microscopic tools for effective diagnostics and drug delivery platform. So today is going to talk about oral delivery of insulin, a mission impossible. So over to you, Professor Sourav.
Thanks, Dalbini. First of all, it's a great opportunity to be here again. I mean, yesterday was a great day. I presented some data on tissue microscopy, and I think today we will just continue with what we have where we left yesterday with my another passion in my scientific career, that is oral delivery of insulin. Now, being a diabetic myself and being on insulin therapy for quite a few months of my life, even more than a year, I would say. I mean, insulin is a molecule that has been involved with my familial history, and I had family members taking insulin, and later I kind of, having carried that legacy, genetic legacy, I have also taken insulin. So I think there is a natural kind of an inclination of finding or being a person or a patient on insulin therapy, and that made me kind of more inclined towards understanding the intricacies of the insulin molecule. Now, I have spent quite a deal of my life or research career, I mean, trying to understand insulin. I don't know how much I have succeeded. My own belief is, at least now I know how little I know. Now, the first important bit that I would like to talk about today, and that's where my lecture or my talk today is based on, is about oral insulin therapy. And where do we stand actually? Now, to understand oral insulin therapy or oral insulin formulation, the truth is that we still have not found that magic bullet, that oral insulin formulation, that we kind of have a kind of a hypothetical situation where the patient will be taking an oral medication in the morning, or there will be some tablet forms. That will be certainly become better from the perspective of patient compliance. But we have not succeeded, or at least our success has not been stellar. And I keep on wondering why it has been so. Because if you look traditionally and even historically, diabetes is a pretty archaic disease. It has been known from antiquity about that diabetes has been recorded in ancient Egyptian texts, ancient Indian texts, ancient Chinese medicine texts. But so far, and by the way, the scientific community is celebrating the centenary year of the discovery of insulin. But so far, it has not been a great success in achieving oral insulin formulation. I will quickly go on. Here's for Yep. Yeah.
Okay. Otherwise it's disturbed the. It's okay.
Yeah.
Thanks.
So now what I'm sharing is, this is an Ebers papyrus. You can find it in the University Library of University of Leipzig in Germany. And this Ebers papyrus, I mean, historians and Egyptologists date it almost to 1550 BCE. So almost 1500 before Common Era. And this papyrus is extremely important. This is because it is one of those very few Egyptian papyri that actually chronicled the different medical treatment facilities or medical treatment modalities that ancient Egyptians followed. Now, what is very interesting is that this papyrus actually also chronicles a disease treatment called, which basically the symptoms were polyuria. And there is no harm in predicting that ancient Egyptians were already talking of diabetes, because diabetes mellitus, of course, is one of the keystones of diabetes, or the symptoms is polyuria. And so it was already known in Egyptian times, and I will not go into a lot of detail what this papyrus actually tells how to treat diabetes. I mean, it was just a mix of some elderberry, some other stuff, and some milk, and that they thought will decrease diabetes. But the time moved on, and this type of antiquity and that kind of medical procedures has sustained in ancient medical literature. Yeah. Yep. Thank you. Sorry. Anything else? Admit? Yep.
Yeah.
Okay. Well, but coming to modern era, it is centenary year. So 1921 to 1922, that was a great era of insulin discovery. And it was discovered by, as we can say, the quartet of Canadian researchers from University of Toronto, namely Frederick Banting. I will start, the Frederick Banting, and then John MacLeod, and then they shared their prize also with Charles Best and James Collip. James Collip did a phenomenal job by actually purifying the canine pancreatic extract that was later used to save the first patient. I will show it here, Leonard Thompson. He was the first patient. He was just then a 14-year-old boy and almost ready to die, because type 1 diabetes was almost a death sentence on those days. There was hardly any medication. And basically, the patients were put on carbohydrate deficient diets. And sometimes the energy intake was given even a meager 450 calories per day. And a lot of those patients, unfortunately, died of starvation rather than diabetes. So this quartet of Canadian researchers actually they took some canine pancreatic extract and injected it to this Charles Leonard Thompson. And that created a beautiful result, and the patient survived. The patient, when he was given insulin dose first, he was 14 years old, and he later died when he was 26 years. But here comes the interesting fact that the patient died after 26 years, not because of diabetes, but because of pneumonia. And that created that historical journey with insulin. And that became, of course, a big news. And this is a paper cutting that actually come out, the Toronto Doctors on Track of Diabetes Care. And you can see of the diabetes sufferers given message of hope. Now that means 1923, there was a Nobel Prize in Physiology and Medicine given to this quartet, Banting and John MacLeod, and they shared their prizes respectively with Best and Collip. And that started the journey of insulin. 1923, Nobel Prize in Medicine. Now, it fundamentally means that this has been a centenary year. And let me tell you that, of course, we celebrate insulin, but at the same time, there has been a century-old, I should not use the word failure, but let's say a lot of disappointment while looking for oral insulin. Because the moment this quartet came out with this news of insulin that can treat diabetes or at least bring down blood glucose, there was already an immediate hype looking for oral insulin formulations, because there were a lot of things going on those days. Of course, some of the sophisticated tools for protein characterization, like X-ray crystallography, was not there, or they were pretty much in their nascent states. High volume industrial scale production of insulin was not there, of course, because there was no DNA recombinant technology that came out at the later part of the 20th century. So there were a lot of issues. And of course, the insulin that were used mostly was porcine and bovine insulin, given parenterally, of course, and multiple times over a day. And that, of course, becomes quite traumatic for the patients, even had quite an impact on their mental health, I would say. And of course, there was deterioration of patient compliance. Now, from that very point of deteriorated or inadequate patient compliance, began the strive towards developing oral insulin formulations. So oral insulin formulation, or a drive, or this journey towards developing it, started immediately after insulin was actually discovered by this Canadian quartet. But so far, not much has been achieved. And I sometimes wonder why it has been so. Before I move there, I'll quickly like to give you this timeline that has appeared, or that we can kind of bring together or converge in the history of insulin's discovery. We started over here in 1921. This is Banting and Best. This was one of the diabetic dogs where the pancreatic extract was given. And that started the process, because dogs were the first experimental animals used by Canadian quartets to start their experimental work. Now, there has been claims that there were other scientists like Nicolae Paulescu and German physiologist Ziegler, who has tried this in the past. That created a lot of controversy, but let's not get over there. Let's stay to Banting and Best. And from then on, there was the first person, Leonard Thompson. I have talked about him already. And gradually, we started understanding what is type 1 and type 2 diabetes. And then in Dorothy Hodgkin in 1969, phenomenal work. She found the molecular structure of insulin. Frederick Sanger, at the same time, was the first person to understand the amino acid, the sequence of protein, the insulin. So there was a lot of things going on with insulin, and insulin has that kind of a moniker, has that kind of a respect in scientific fraternity, that it is associated with many firsts. It is the first protein to be completely sequenced, first protein to be X-ray crystallized and looked for most of its structure, its crystallized structure. And this is fundamentally also very important. To the best of my knowledge, insulin is the only protein that is produced in human body in crystallized form. That's incredible. That's usually not found. So there are a lot of interesting things. Insulin was also the first protein which was created by DNA recombinant technology. So there are a lot of firsts associated with insulin. And that makes this not a failure, as I say. That's kind of a disappointment. Always looking for oral insulin, kind of more painful, because we know about diabetes from antiquity. We know about insulin pretty well. This is the first protein molecule where it acted as a template where we tried to understand, and we succeeded in understanding a lot of things about mysteries of protein in general, but we are still struggling to find an oral insulin formulation. And today I'm going to make a case, and I'm trying to convince you, where I will ask you to seriously reconsider that is it fair or is it pragmatic now that we even chase the dream of oral insulin formulation? And that's where I will put my topic over. Now, if you look today, if you go if you go for the typical diabetics, how they are treated, parenteral insulin formulations are all that you will get in the market. Of course, you will have these insulin pumps and insulin pens. I have myself been taking these insulin pens for quite some time now. My father also takes them. I mean, there are incredibly developed technologies now. I will just like to take your notice to this dial in insulin pens, where you can actually precisely load the pen with the amount of insulin you need or the patient needs. And that amount of insulin that the patient needs is usually decided by a sliding scale that is prescribed to the patient. So there are advanced glucometers now where you can measure the capillary blood glucose ASAP immediately. And then as per the sliding scale, the patient can upload or patient can dose the pen, and then inject it, often self-administered. And this dial even gives a click noise. It clicks when the dosing is finished, and the patient knows very well that yes, the amount of insulin required given to the sliding scale of blood glucose levels has been delivered subcutaneously, safely, and fully. So there are a lot of developments going over here. And I think especially over the last 5 decades, this microneedle technology has made so much of advancement that many of these injections that the diabetes patients has to take multiple times daily are almost, if I say, I might even say, they actually has become painless. And then you are coming with the modern AI technology with all these insulin pumps that gives a 24/7 monitoring, and it is rest assured that given the situation or given the amount of blood glucose, this pump will deliver the insulin as per the requirement. So both the patient and the physician, they are assured that enough insulin will be delivered to the patient depending on the fluctuation of blood glucose or this diurnal variation of blood glucose. So there has been a lot that has been achieved already. And that's where I would like to know why oral insulin is such a big problem. My own understanding is that if we want to dig into this problem and try to understand the reasons for our failures, we need to understand the insulin molecule first. And it is my own feeling that we are too inclined towards developing an oral formulation rather than trying to understand the insulin molecule itself. Because whatever formulation we might develop, at the end of the day, we have to deal with insulin molecule. And that is where the issues begin. Insulin itself is a very strange molecule. I think it's a magic of nature. Only nature could have created a molecule like this. If you look carefully, and by the way, I'm only here going to talk about human insulin, but the amino acid sequence of insulin is actually greatly preserved across species. And that's what we will see. And that is why, in fact, you will be surprised that insulin is also present, or insulin-like molecules are also present in other forms of life. Example, fishes, example, invertebrates. And what is also very interesting, that the amino acid homology or the similarity between human insulin and fishes is also startling. And some of the invertebrate insulin or insulin-like molecule, that is also the amino acid sequence is pretty conserved, and they're pretty similar to human insulin as well. This is one of the reasons insulin is also one of the molecules which can act cross-species-wise. So porcine insulin, bovine insulin worked well in humans, and before the DNA recombinant technology arrived, they were the life saviors for human species, for Homo sapiens. And in fact, there are evidences emerging now that even insulin-like molecules present in piscine species, they even are able to bring down glucose in human cells. So there is an amazing, incredible spectrum of this cross-connectivity of different species by this one single molecule. And that is exceptionally a mysterious thing. And that is one of my understanding, and that is where I get sometimes confused how this is even possible, that there is one molecule which has many variants, and that's spread across almost the different life forms. In fact, my understanding is that insulin molecule appeared in life cycle or in this world, in the forms of life, maybe billion years ago. And the current hypothesis is wherever the metazoans or metazoon animals or the species appeared in this planet, insulin appeared as well. So anything you are dealing with a metazoa, you're somewhere, there has to be an insulin or insulin-like molecule. Another interesting fact about insulin. Insulin is part of a family of proteins called insulin-like growth factors, IGFs. And this IGF consists of many different family members. But of course, the most insulin part is, or important members are insulin, insulin-like growth factor 1 and 2. And there is another protein that is very important that is called the relaxin. And this relaxin, it's a hormone. All of these are neuroendocrine hormones. And relaxin actually helps in the relaxation of uterine muscles during parturition. So there is a diverse range of different neuroendocrine hormones that belongs to this family of molecules, very intricate molecules, and insulin is just one of them. Now, for whatever reasons, very mysterious, very unknown, that in this evolutionary cycle of life, insulin actually was given the workload of metabolic control, whereas the insulin-like growth factors, they still keep on working focused, or they stay focused on the growth. By the way, insulin can also have certain growth factors related to it. Insulin can still promote growth. There are enough reasons to believe that. There are experimental data. But for whatever reasons we still don't understand well, insulin also has that additional workload of metabolism. It plays a huge role, and that is why diabetes has emerged as a metabolic disorder. Let's look into the molecule of insulin. Insulin molecule, again, I'm talking of human insulin with mild variations. This structural template remains same for all the other insulins, including bovine and porcine insulins. Human insulin, the molecular weight is around 6 kilodalton. It has two amino acid chains. One is the A chain, another is the B chain. A chain is consisting of 21 amino acids, B chain 30 amino acids. So altogether, insulin has 21 plus 30, 51 amino acids. The linkages are shown over here. There are two interchain disulfide bonds. One is between A7 and B7, and the second one is A20 added to B19. Other than that, there is one intrachain disulfide bond, which is over here. You can see this intrachain disulfide bond that actually make this beta helix possible. What is interesting is there are a couple of things I would like to point out, which I think is very important to understand the physiological or biological behavior of insulin and why it is difficult to manipulate this molecule beyond certain degree, which we need to achieve if we have to find an oral insulin formulation. The number one is, if you look to the C terminus of the B chain, it is extraordinary. Extraordinary in its way, because you now have a 10 amino acid which are linked to each other, and they all are hydrophobic in nature, which means the C terminus of the B chain, this patch of 10 amino acids, they're highly hydrophobic. And this enables insulin molecule to have a lot of unwarranted hydrophobic reactions with a lot of different materials and surfaces. And these hydrophobic reactions is something which is often working as a hindrance while manipulating insulin or even encapsulating them in between or within, say, for example, nanoparticulate matters. Nanomedicine, which we believed, or at least one time we believed, that will enable us, it will be an enabling technology to overcome certain barriers. For example, oral insulin delivery. This is one of the issues because the different materials, for example, polymers, which you are trying to make the nanoparticles, which probably some encapsulated insulin, this hydrophobic patch in the B chain, in the C terminus, of course, that quickly triggers a lot of hydrophobic reactions, and then it creates all sorts of unwarranted binding and different other modalities with changing in the configuration of insulin molecule. So that is already a huge problem. We don't know how to deal with it from a material perspective. There are some other points I would like to draw your attention. Now, these black or these darkened amino acid residues, they're conserved across species, human, cows, pigs, and there are more. So these species, these darkened, these residues are, they're preserved across the spectrum of different species. These are the dimers. These amino acids, which are marked with D, they are the residues that actually help or trigger amino acid, the oligomerization of insulin. So for example, this glycine over here, these amino acid residue actually partakes or helps insulin forming dimers. Then there are residues which I have marked with capital H, that H means hexamer. So these are the amino acid residues which helps hexamerization of insulin. Now we are already talking of dimerization and hexamerization. In short, oligomerization of insulin. And that is another kind of a challenge while working with insulin. Insulin is a strange molecule in a way that it can form different oligomers. It can conglomerate together multiple insulin molecules, and that forms certain agglomerates. Now, What is also very mysterious about insulin is that insulin is produced in pancreas. It comes a long way through cleavage and certain maturation and engineering process. So it is produced as pre-proinsulin. That pre-proinsulin is further modified into proinsulin, and that is further modified into insulin. Now, insulin, if you look into the molecule, that is preserved as a hexamer inside the beta cells of the islets of Langerhans located in pancreas. This hexamer is a typical kind of a nanoball or kind of a nano construct, and it has a nano cavity which is held together by 2 zinc cations and 10 water molecules. These 10 water molecules provide further stabilization to this hexameric insulin by providing hydrogen bonding. So 2 zinc cations and multiple hydrogen bonds provided by 10 water molecules that hold these 6 insulin molecules together to form this hexamer. The interesting bit is this hexamer is exceptionally stable. It can stay there. And the important, again, and another piece of information is that all these insulin molecules, while in hexamer, they retain their physiological activity. But if you just inject the hexamer into the blood, it is inactive. So oligomeric forms of insulin are physiologically inactive. That means they will not bring the blood sugar down. To bring the blood sugar down, actually, the hexamer has to fall apart. The oligomer has to unpack into lower unit monomers. So I have just shown here, in x-axis, these are the insulin oligomers. And of course, it shows the stability as well. And on this axis, I have shown the energy because I'm trying to understand these different oligomeric forms from a thermoenergetic perspective. Now, if you look into the hexamer, this is how 6 insulin molecules come together, held together by 2 zinc cations, and that is how they are preserved in pancreatic islets. They're very stable. In fact, this hexamer has 2 different forms or conformations. One is a little bit tensed, so in the energy profile, it is slightly more energetic / reactive. And there is a relaxed state, which is over here, which is hardly any reactive. It's very stable. And both of them actually don't bring insulin down, so they are physiologically inactive. The magic starts when there is an increase or spurt of blood glucose, say, for example, after meal or after the patient has taken some sugary drinks, for example, some fizzy drinks from some Coke or something. And whenever there is a spurt of blood glucose, the sensors sense, of course, they sense it, and there is a trigger of releasing insulin. So what happens then is this hexameric insulin gradually unpacks into dimer, trimer, they can even be tetramers, and finally, it attains its basic monomeric form. This monomeric form, which is quite reactive compared to if you compare to the hexameric form, this monomeric form actually comes into the blood, and that monomeric form is what brings blood glucose down. So this is kind of a dilemma already. Insulin is preserved as a hexameric form, but it is not physiologically active. But it is active only as a monomeric form, but the monomeric form is energetically quite active, and this monomeric form is the reason also for agglomeration. And insulin agglomeration has been one of my research interests because not a lot has been done on it. And my student, PhD student, Meg Ren, if he's watching over here, he has done incredible amount of work towards understanding insulin agglomeration. And when we are talking of insulin agglomeration, this is where it all starts. The monomers are pretty thermoenergetically active, and they quickly get into agglomeration and binding to each other. Not to forget about that hydrophobic patch that I talked about in the B chain on the C terminus of 10 amino acids, that also takes a lot of part in different hydrophobic interactions, which are often unpredictable, and you cannot quantify it. I will now get into the real bargaining chip here. That's the why oral insulin is such a difficulty. And I will try to kind of flesh out my point based on 3 barriers. I think these 3 barriers are our main obstacles towards gaining or getting or towards achieving an oral insulin formulation. And I gave a kind of an introduction to understand insulin molecule because we can correlate a lot of these challenges with that insulin molecule, these intricacies of insulin molecule. We need to understand the molecule first before we try to understand the challenges, and even before we try to even find a solution. And that is why this introduction to insulin molecule was necessary. Now, the first barrier, first of these three barriers that I want to talk about, which is an obstacle that making oral insulin formulations almost a mission impossible, is the fluctuating pH within human gut. Now, human gut, again, a very long pipeline ductular structure. It starts with, again, I will not go into a lot of anatomical jargons over here, but of course it starts with the oral cavity, then esophagus, which finally culminates into with the stomach, then goes with duodenum, jejunal coils, ascending colon, transverse colon, descending colon, etc. And this cartoon looks great. But if you look into the pH, that means how much allowance of protons, or how much protons are present, human gut has a wide variety of pH. So for example, the stomach and the bottom part of the esophagus, when it is connecting to the stomach, that's highly acidic. And this highly acidic because of preponderance of hydrochloric acid. That means there are a lot of protons, and the pH in stomach can sometimes even fall up to 2 to 3. So that's quite acidic. And that's a major challenge because any oral formulation of insulin has to somehow withstand this assault of acid. And it's quite a bit of hydrochloric acid over there. And protein molecules, given its secondary structures, are always almost vulnerable to this acidic insult or acidic assault. Now, that's one of the biggest challenges, that if we have an oral insulin formulation, how to actually go through and sustain this acid within stomach or gastric acidity. Now, another important point is, if there is an oral formulation and if, hypothetically, we are able to cross this stomach, then even throughout the rest of the gut, the pH keeps on varying. You have to measure or you have to match this color scale with whatever you find here. In fact, in jejunal, the pH can even be neutral, 7 or even 7.2, which is physiological pH, I would say. But so this pH or how much protons are present, it varies across the length of the gut. And if we are having an insulin formulation, first it needs to somehow sustain its secondary structure, which is very pertinent to preserve its biochemical perspective or physiological activity. That needs to be sustained first. And that's already a big, big challenge. We don't know how to circumvent it. And even if it passes through it, there are different sections of the gut with different pH. They're all trying to influence the insulin molecule in different other perspectives. And the insulin molecule has to sustain this rat of different pH atmosphere and then get absorbed. So that's quite a bit of a challenge for insulin molecule when delivered orally. That is, in my opinion, a big, big challenge or the barrier number one. And when you put, and again, in collaboration with this group over in Estonia, my student Megrain and I have done a lot of interesting investigations over the last couple of years, trying to understand these different models of insulin agglomeration. So for example, we were talking of the pH or very acidic pH in stomach, which can even fall up to 2 to 3. Now we procured some human insulin that is easily possible these days. And we subjected this insulin to the pH that we typically get in human stomach, which is around pH 3. And this work was again done by Chanaka over here, where we did a lot of NTA studies. I'll show you the data in a moment. But what you will find is if you subject insulin molecules, and I'm particularly talking of human insulin molecule that we procured from Sigma Aldrich, and if you expose them to a pH 3 buffer, this is how you will get. So this is the transmission electron microscopy of the insulin agglomerates. So these are insulin agglomerates. The scale bar is 1 micrometer. And you can clearly see now this is a broad field view of the different insulin agglomerates. At least what we have found, the agglomerates were kind of spherical. Now, if we focus on just one single insulin agglomerate, so this is 100 nanometer scale bar, this is how it looks or appears. You have a centralized kind of a core where the molecules are kind of more jumbling up. They're much more clustered, while as you move towards periphery, they look much more loosened up. Now, in a transmission electron microscopy, this type of a dense core that is gradually loosening up or unfastening towards the periphery is not completely unexpected. And we roughly have found this by other studies as well, that these spherical insulin agglomerates coming due to acidic influence or acidic contact are roughly 400 to 500 nanometer in diameter. And they are, I would say, at least for our samples, they were pretty monodisperse. So there was not a lot of size variation. They were pretty stable, colloidally very stable as well. And we could get this fantastic transmission electron microscopy figures. Then in end of 2022, we were supposed to, I did some NTA data, NTA nanoparticle tracking analysis with Chanaka sitting over here. And we could even see that light diffraction or scattering from these insulin agglomerates. So that these bright spots that you are seeing, these are colloidally stable insulin agglomerates moving in a suspension and the light being scattered by them. So we could do even light scattering experiments, which was great because typically light scattering experiments are done well with metallic nanoparticles, but these colloidally stable particles were good enough and we could do the scattering experiments as well. We measured the hydrodynamic diameters of these insulin agglomerates as well as the zeta potential, and I would say they pretty much agreed with what our findings of transmission electron microscopy. We did some fluorescence lifetime imaging microscopy. I don't have time to go into a lot of details, but we actually got the insulin molecule, the same human insulin molecule, which are now conjugated with the dye FITC, fluorescein isothiocyanate. The interesting point of fluorescein isothiocyanate is that its fluorescence lifetime varies with acidity. So it's in normal pH or in neutral pH, the fluorescence lifetime of tau of FITC is around 4 nanoseconds. But when the acidity increases in the biological milieu, this fluorescence lifetime actually becomes less, it falls. On the contrary, if the pH is more, that is, it slides more towards basic scales, the lifetime of FITC increases. So fundamentally, this FITC level insulin can be used to understand these agglomerates based on using FITC as a molecular pH meter. And that's what we did. This is a wide field of insulin agglomerates. This insulin we procured from Sigma. They were conjugated with FITC, and the rough ratio of conjugation was 1 FITC molecule attached to 1 insulin molecule. So the molar ratio was 1:1. That is how the degree of conjugation were. And these are huge variations. We see a different agglomerates. I have particularly pointed out 3 regions, region 1, region 2, and region 3. Before I go into more detail, I would like to show you, this is the color scale developed by the software, and this is where the fluorescence lifetimes are given. So in short, so we actually measured, or we actually designed this color scale varying from roughly 0.187 nanosecond to 0.298 nanoseconds. So I would say roughly 0.2 to 0.3 nanoseconds. That is where the lifetime of this FITC molecule attached to insulin varied. And just to let you know that the more the color scale goes towards blue side, that is more acidic because that's the falling lifetime. So that means that area or that zone is more acidic. And while it shifts more towards the red zone, that is where it becomes less acidic / more basic. Now we have fundamentally found out 3 different, now there are more regions, but we have arbitrarily chosen 3 regions or 3 larger agglomerates, region 1, region 2, and region 3. And on the right bar, I have kind of stretched out these 3 regions. So this is region 1, region 2, and region 3. Let's look closely into the agglomerates. You will find there are different acidic zones within the same agglomerate, by the way. Those are marked by the blue regions. And that gradually kind of spreads out towards the periphery, which are more into the reddish, and some yellowish zones are also possible. So if you move by this color scale, that means the lifetimes increased. Same for the other region 2 and region 3 agglomerates. For example, if you look into the region 3 agglomerates, it's clearly visible there are blue regions, which means hyperacidic regions within the agglomerate. But at the same time, when it goes towards the periphery, it is becoming gradually, the pH, the acidity is decreasing, and it's gradually transitioning its color to yellow to certain more reddish zones as per this color scale as well. So that means within the same segment of agglomeration, we are finding different regions of acidity as well. And what we have always found that the acidity is more in the core. Now we can discuss it why it's that things happens during the Q&A session, but this is a very consistent finding in all our data. This is again, we are very proud of this image because this is again the advancements that we have been able to get into light microscopic techniques. This is an individual insulin agglomerate where you can see there is a hyperacidic zone with this blue one, and that gradually transcends into the less acidic zones. You will see gradually the color changes. It's almost like an onion peel. The layers are being deposited on top of each other, and that is gradually transition across the scale bar gradually towards the less basic or less acidic or the more basic zones. And this is 100 nanometers. So this agglomerate was roughly 300 to 350 nanometers. And this is the prowess of modern light microscopic platforms that can enable you to get resolution up to this extent. And not only that, you can even see the different layers of acidity by the virtue of having FITC attached to that insulin molecule. And by developing this molecular pH meter with the help of fluorescence lifetime imaging microscopy. I think we are pretty convinced that this acidity does cause insulin agglomeration. So how to circumvent it, we are still pretty much, we are still pretty much confused about it. One of the things that can be done, I'm coming back to that if we can encapsulate insulin within some pH sensitive material, for example, polymers. Before I even go there, I will quickly like to touch upon the recent developments in my lab, where Megrain has started doing experiments that, okay, acidity is already causing agglomeration. What about temperature? Insulin is also like many of the proteins, for example, they are pretty sensitive to temperatures as well. So what I will not go into a lot of detail, but I will just give you some nuggets of information that we have been recently getting from our lab, and we will be delighted to collaborate. Again, I'm here to do some NTA experiments with Chanaka, and I'm pretty sure these results will be great to publish somewhere end of this year or maybe early next year. Now, what happens is, so this is, for example, this is insulin in pH 3, the concentration is 0.25 mg per ml. We have heated it up into 37 degrees centigrade, 48 hours, and we do see spherical agglomerates, which is what we expected, because 37 degrees centigrade is pretty much a physiological temperature, so it should not do anything. However, when we actually heat it up to much higher degree, 65 degrees centigrade, we see charring already happening. Now I am not showing the data over here, but we have done a lot of experiments with circular dichroism, where we definitely see the secondary structure of insulin is also completely disrupted. So 65 degrees centigrade kind of is the higher threshold / positive control that will take out the secondary structure of insulin, which means we securely interpret that more. So sorry guys, again, some edodome issues, connectivity issues, but we have restored it. So I will pick up where I actually left. So we were talking about the transmission electron microscopy of the different insulin agglomerates. I have shown you that when heated to 37 degrees centigrade, which is the physiological body temperature, not much happens, but we still see agglomerates at pH 3. When we heat it up to 65 degrees centigrade, the agglomerates are much more bizarre looking. And in fact, I am not showing over here, but I have circular dichroism CD data to show or to demonstrate that the secondary structure of insulin was completely gone by that time. The typical stretches that you are habituated to see in the CD spectra of insulin, that is almost all gone. So by this 65 centigrade, that's kind of that upper threshold or / positive control. That is where the secondary structure of insulin are completely perturbed. And what happens is, again, I did some experiments with my PhD student with FITC level insulin, and that created a lot of agglomerates when heated to 50 degrees centigrade, and we heated it up to 72 hours. I think we can clearly see the different FITC level insulin agglomerates. The scale bar is given here, which is 500 nanometers. And these agglomerates are pretty much spread around, and we could even see there are different variations of lifetime. If you look carefully into individual agglomerates, you will see the color or this color scale that is pretty much fluctuating within individual agglomerates as well. Now, because these agglomerates were much larger, we could even do zeta stacks and create 3D visualization in hyperspace. So this is one of the insulin agglomerates which I have created, the 3D visualization. The concentration was 0.125 mg per ml. It was heated to 50 degrees centigrade, and we continued heating up to 72 hours. And this is what we actually find. So I will rotate it. So this is how this 3D structure of the insulin agglomerate looks like pretty much. You can see there are still different levels, different segments of acidity. It's different segments of acidity over here. And if we move on again, we can see the different structures of insulin agglomerates. I will move on. We have more data about it. So this is insulin concentration 0.5 mg per ml. We heated it up to 65 degrees centigrade, up to 72 hours. And that should have created, as we know, heating up to 65 degrees centigrade it is supposed to disrupt the secondary structure, create agglomeration, and we have seen that. What we also see at 65 degrees centigrade, that these insulin agglomerates not only just produce agglomerates, but they kind of attach to each other, causing this kind of a wave or network. And I could also create the 3D structure over here, as we will notice that they're kindly cross-linked structures, which I would expect in case of a disruption of the secondary structure of insulin. Now, barrier one, I think I have convinced, or at least tried to convince you, that what are the issues with oral insulin formulation. Now, I will actually move on to the second barrier, which is
We cannot see you.
Yeah, that's fine. Now, I will move on to the second barrier, which is the gut mucus barrier. Now, due to crunch of time, I will not go into a lot of details with the gut mucus. But in simple terms, I would say the gut mucus is a hydrogel structure which protects and bathes the inner luminal surface of the gut pipeline. And it actually protects not only the gut, but also our body physiological system from the assault of different toxic materials from outside. Now, this gut mucus layer, this is spread across not only the stomach, but also throughout the small intestine and to a certain extent of the colon as well. Unfortunately, the gut mucus layer is not very permissive towards the different nanoparticle formulations. If you remember, this stomach acidity is one of our barrier one, that is the first barrier that any oral insulin formulation has to negotiate. Then how shall we negotiate? One of the hypotheses that emerged was that if we can encapsulate insulin as a drug cargo inside a nanoparticle construct, or it might not be also always nanoparticle, but within a particle construct, which might be nanoparticle, and then it can preserve the insulin from the wrath of gastric acid and flow with the peristalsis towards the small intestine, maybe duodenum, jejunum. Jejunum is the most optimal site for delivery. And there are places called Peyer's patches within the jejunum where the mucus layer is deficient. And it is through these mucus deficient Peyer's patches, that is where these particles most probably can come close to the gut epithelial wall. And we still don't know the picture, but probably some of these particles will already dissolve by then, or some of the particles will hopefully be able to cross the gut epithelial barrier. We still don't know what's the exact picture, but most probably the realistic interpretation is that it's a mix of all these different scenarios that can happen when the nanoparticles actually come close to the gut mucus wall. We still don't know. Unfortunately, that is not always a success. The mucosal barrier that we have in gut, that is pretty robust, and it has evolved over epochs. It is created by nature as a very formidable, as a formidable physiological barrier. I will move it here. Yeah, as a formidable physiological barrier to stop different materials from accessing the epithelial layer. The mucosal barrier has different layers as well. It has a first outer mucus layer, which has a lot of biota within it. Then there is inner mucus layer, which is more protected zone. And then this is the epithelial layer. So what happens is, if you are putting some particles, which may be nanoparticles with some encapsulated insulin, and say, for argument's sake, this particulate formulation has sustained its ability to deliver insulin even after going through the acidic milieu or the gastric acidity. If it comes in close to that, these nanoparticles are often trapped within the mucin network. This mucin is a fibrillar protein that makes the major solid component of mucus. They can carry water, they can swell up, and that creates a mesh of mucus. Now, what happens is, if you have particles, some of the particles can freely diffuse, although that is rare. Some of the particles will be actually coming over and they will be repulsed due to steric hindrance. And some of the particles will just be trapped out. So even if we can put some nanoparticles carrying insulin into the small intestine, say, for example, jejunum, this mucus layer, which is the second barrier, is a very difficult barrier to negotiate. So first, we have already a barrier 1 with gastric acidity. If we cross that, we have the second barrier, which is the gut mucosal barrier. And these are the different intestinal villi. So there is a huge amount of surface area present in the intestine, which is, again, this is just a schematic, and the mucus layer is not shown. But there's a huge surface area in realistic terms that this nanoparticles comes in contact also with the mucus layer, where it actually has to negotiate. So this second barrier is also quite extensive. Now, what if, this is my third barrier, what if, even if the particles cross the two barriers, it has crossed the gastric acidity barrier, it has crossed the mucosal layer barrier, mucosal physiological barrier, and let's hope it is absorbed in the gut. But then comes the third barrier, and the third barrier is that any particulate material that is absorbed into the bloodstream through the gut, that is immediately diverted to the human liver or the hepatic system through this hepatic portal vein, which carries basically 80% of the blood flow to the liver. So most of the administered dose, whatever we could actually transfer across gut mucus after crossing these two barriers, most of the dose, sometimes more than 95% of the dose, immediately sequesters inside the liver and metabolized. So after these three barriers, I will presume there is no percentage of the oral dose left, or whatever is left even, that is too little to deliver any therapeutic impact. Nonetheless, if you are going on the nanoparticle route, nanomaterials are toxic by their own ways. For a disease like diabetes, where strict dosing control is of paramount importance, can we really afford to provide or to dose the patient with so much of nanomaterials which are toxic by their own means? I think there are serious concerns now. And it is my view that despite knowing a lot, we should really think, shall we perceive or shall we proceed this way of oral delivery system? Because we know what are the barriers, and unfortunately, we still don't have a foolproof, optimized, and globally acceptable way or protocol to knowing how to negotiate with these barriers. And unless we can negotiate with these barriers, in my opinion, the dream of having an oral insulin formulation will never be achieved. And that is where I'm trying to make. If you look conversely, the parenteral formulations, they have made great strides ahead. The needle technology has changed. With the advancement of digital technology, now we have insulin pumps. And most of the issues that were hurting patient compliance with parenteral formulations has been addressed or have been largely addressed. More importantly, a lot of these barriers that we surmount or we face in oral insulin delivery systems are simply not there because these are parenteral systems. That means the bioavailability is way higher, it is way more controllable, I can measure the dose, and I can give the precise dose, which is of paramount importance in case of diabetes, because strict dosing control is what we need in case of diabetics, especially in modern day life, which is rushed, and there are a lot of lifestyle issues as well we have to handle with. That is what my point is. Everything is possible in science. I don't believe oral insulin formulation is impossible. But my point to the scientific community is it worth chasing? Because we have already the issues with parenteral formulations, which has a lot of advantages as well, including precision in dosing, that has been already addressed or to be addressed. But these three major physiological barriers, at least I don't see any major strides being made. If we keep on chasing this pathway, we will be, of course, wasting taxpayers' money, funding, but most importantly, probably we will be wasting a lot of animal lives doing animal experiments, which is ethically incorrect. I'm not saying stop it completely. I'm saying let's know how to negotiate with these barriers. Unless we can know how to negotiate these barriers, just giving fluffy rhetorics will not work, and we will be moving in circles, which I think over the last century we have been. So this is a warning sign for me. I will thank a lot of people. Now, there is a lot of people to thank, so I have basically clustered them in my UCD research for funding. Of course, money is important. My supervisors, and I know Professor David Braden is present here. So David, if you are listening, you kind of baptized me in this field of oral insulin delivery, and I want to say that I'm really grateful and appreciative of what you have done for me. And of course, all other supervisors, my colleagues, a lot of them, which I work in Transient during my postdoc times. There has been a lot of simulating discussions, and I have learned a lot from them. My students, I don't know even how to start. Megren, my PhD student, if you are listening, I'm proud of you. You have done a great amount of work. There are other students who have worked in this project. Zuzana Grenschuk, incredible work. Salome, if you are listening, your work has been great in understanding how temperature variation actually influences insulin agglomeration in turn. Other than that, I'm very grateful to Ali Raza Fazili's lab, who has hosted me here. Chanaka is sitting here. He has done incredible work with all this NTA stuff, GetNet, you as well. I think this NTA data has enabled me and us as a team to understand the intricacies of insulin agglomeration, how these agglomerates actually behave, and they never stop actually surprising us. Finally, I would like to end my presentation, and I would like to take any questions if you might have. Thanks for your time. Thanks for joining online, and apologies for the connectivity glitches. Sorry. Thank you. Any questions, please? Yep. Yep, you have to sort it out.
Sharing this. Yes, Ali Reza, please go ahead.
Thank you very much, Saurabh. It was really interesting to listen to you. You are a very good speaker. Definitely, you going from the history and coming back to the thing. One point, maybe I don't know. We are working nowadays a lot on milk extracellular vesicles, and what is amazing about them, they go through the same route that you have tried to go with your milk with your insulin, but they are able to sustain the pH. I mean, the stomach pH, and with the experiments people have done, we haven't done, but they can somehow deliver their cargo after being somehow orally administered, and they can somehow sustain the stomach pH and then get probably also to the intestine. I don't know how it would be when it reached to the liver, and it was also interesting for me to hear your work on the liver. But what do you think? Is there a possibility, for example, to package insulin in the milk extracellular vesicles and use that as a way of somehow delivering it to the patients, or from oral point of view? I do know if anyone has tried to do something like this.
Thanks, Professor Fadili. Thanks for your kind words. I hope I have spoken not very badly, and thanks for your appreciation, actually. Listen, I am always intrigued with exosomes, and when I have used the word nanoparticles here, I have fundamentally meant nanoparticles which were synthetic. That means they were probably polymeric nanoparticles, which, I mean, with great strides made in polymer science over these days, a lot of polymers and different other materials, nanoconstructs have been synthesized in the labs. Exosomes are a different species, though, because these are indigenous particles. And my understanding is, again, I don't have data, but I have a hunch. And my hunch is that probably exosomes are better dealt in human body, because if you look into this mucus layer that is in the gut, it has basically a good book and bad book. So some particles or some materials, it actually lets to pass through it. Even the mucus layer lets it pass through. So it lets it permeate. And some particles, it just tags foreign, antigen, pathogen, I don't know. And it doesn't. So I think exosomes can be a better bet. There are a lot of issues with it. Now, exosomes also come with certain downsides as well, because I think any oral formulation, especially if we are going on a particulate route, any oral formulation of such particulate material has to be very or quite monodispersed. Now, exosomes vary a lot over their dispersion. So that might be one of the issues. But I personally think exosomes can be a good bet to see if they are better particles, because they are indigenous particles, and probably the mucus layer and our body immunity system as well, they will be dealing with it as more kind of a homely guy. So less of an antigen, more of its own persons. But then again, loading insulin to exosome might not be that easy. We have to do a lot of stabilization assays. We have to see that this, and again, if we are looking from a product perspective, we have to understand that making a formulation is not only enough, it has to have a good shelf life. It has to be cost effective. There are many other ways. For example, Exubera, which was a nasal insulin formulation. It was a useful formulation, but it failed because there was no acceptability on patient's behalf. So the patient also has to accept it. And because if there is no acceptance, that is why Exubera was taken out of market now. It is not available in circulation. So exosome can be a bet, but at this stage, I will be cautiously hopeful. It can work. It can have certain facilities attached to it, which probably a synthetic nanoparticle or particle might not have. But then again, I am not aware of people doing it with insulin at least. If you have some data and if you want to send some samples, we can try certain things. But I think, again, this is completely my hunch, my gut feeling that exosomes, because they are indigenous particles, might actually work better or might actually give us better results. I don't know. And by the way, exosomes are much less toxic as well. So that's already takes out that toxicity perspective or toxicity hurdle of synthetic nanoparticles, which definitely is a plus. I have no idea at this stage, but as I say, I remain cautiously hopeful.
Thank you very much.
Thank you.
Thank you.
Yeah.
Get it? Different layers, yeah. Layers, yeah.
Do you know how much has been like passed through from, for example, from acidic pH, then to mucus, then the last was to paper, but do we know how much has been from each step?
Get it? The issue with this is a very nice question, but the issue is how to know it, because these ratios, how much will actually reach, this is vulnerable to a lot of variations. This is varying. First variable is the formulation variable. Now, let's say we go by nanomedicine route, we have a nanoparticle formulation. And nanoparticle formulations are notorious for batch-to-batch variation. So you give a batch today, the next day the batch will be different, it will be behaving different. And maintaining this homogeneity through batch-to-batch in an industrial scale, I will be very, very confused or not very hopeful about it. Okay, let's presume we have got it. Then again, there are individual variations. When you are dealing with patients in a large cohort, there are individual variations which is often unpredictable. So how shall we even different people have different pH in stomach? They're acidic, but that's not exactly what it is. So what we are asking actually, if we are looking for this proportionality, that we want to know exactly how much, for me, it's impossible. There will be always a standard deviation. And that already makes it even more hard, because how will we calculate the dose? If we are using injection, for example, insulin injections, I know how much dose I have loaded in it and at least how much I have put at it. Okay, even there might be some variation, but the variation is low. But when you're dealing with oral insulin formulation or any formulation per se, I would say, if you're going by oral route, you're going through these barriers, which is fundamentally you cannot model. And if there are sample variations, there are individual variations, barrier variations. And then of course nanomaterials are notorious for all these issues that comes with it. Batch-to-batch variability, even in vitro results don't match with in vivo results. And even if your in vivo results work, you're working with small rodent animals, which is 200 grams maybe. And then to converge it and to expand it to a 70-kilogram human being, that will be very different. So no, I don't have a precise answer how much proportion actually reaches, because there is no specific answer. There is no specific answer. Yeah.
Second question. Like, how do you come up with this ratio, like 1-to-1 ratio? I think it's really regarding
Which one?
Epithelium.
1-to-1 ratio.
That's how, like.
Oh, you mean the insulin that was FITC?
Yeah.
That was supplied, we didn't come out, it was supplied by Sigma. So it's in the production data sheet that insulin was labeled to FITC and the ratio was 1:1.
I thought like you.
No, that would have taken a lot of time and Megret would have still struggling with his first article by now. So because it's not only about conjugation, after conjugation, you have to purify. Because there will be skeptics, they will say, oh, this is not insulin, this is only FITC which is coming out. So no, these are stable FITC conjugated insulin, and the ratio, what I've been told by Sigma is 1:1. It's in their MSDS data sheet. So I have not conjugated it myself. Yep.
Is there any more questions to Professor Sourav? If no, thank you very
Okay.
much for joining with us. And we have a small token of appreciation for
Oh,
your
thank you.
certificate. Chanaka will hand over that.
Chanaka,
and,
okay.
Yeah,
One certificate? Okay.
yes, that's because the previous one was in PMU and this one is in UT.
Okay. Thank you. Okay. Thank you. Thanks, everyone. Thanks for joining. And again, sorry for the glitch on the connectivity. Not my fault, but hopefully it didn't bother you too much. Thanks for the, sorry for the inconvenience. Thanks. Let's stop.
Thank you,
Thank
everyone.
you.
And let's join in our next talk in next week. Yeah, see you then.